Okay, today this video we are going to discuss the emergence of bioinorganic chemistry and the role of metal in biological system. Okay, this video we are going to discuss the emergence of bioinorganic chemistry and the role of metal in biology, functions of biology and in toxicity, etc. Okay, then we are just go through the introduction of this emergence of bioinorganic chemistry. Okay, organic chemistry at one time was thought to be the chemistry involved in the living system because compounds of carbon were found to be playing the key role in all biological process. Okay. However, slowly it was realized that metal ions play a vital role in vast number of widely differing biological processes. Okay, some of these processes are quite specific in their metal ion requirement in that only certain metal ions in specified oxidation state can be fulfill the necessary catalytic or structural requirement. Okay, a large number of metal ions are involved in biological processes. This can be verified by just going through the protein data banks. Example, in Brookhaven protein data bank, 52% of protein contain metals, excluding weakly bound metals such as sodium. Okay, this count include protein and enzyme with heme and chlorine group and unmuted form of certain metallobiomolecules and structure of the same enzyme with different substrate and inhibitors. Okay, metal ions play essential role in about 1/3 of enzymes. These metal ions can modify electron flow in a substrate or enzymes, thus effectively controlling an enzyme catalyzed reaction. Okay, they can serve to bind and orient substrate with respect to functional group in the active sites, and they can provide a site for redox activity if the metal has several valence states. Okay, without the appropriate metal ions, a biochemical reaction catalyzed by a particular metalloenzyme would proceed very slowly, if at all. Okay, so bioinorganic chemistry comes into play. Bioinorganic chemistry encompasses a variety of disciplines ranging from inorganic chemistry, biochemistry to spectroscopy, molecular biology and medicine, etc. Okay, this field is undergoing a phase of explosive growth partly because of exposure and insights obtained by a large number of X-ray structure of several metalloenzymes. So, bioinorganic chemistry is one of the emerging explosive branch in chemistry. Okay, bioinorganic chemistry is beginning to emerge as a major discipline as can be judged by several published textbooks. Okay, then next we are going to discuss elements in body and the requirement. Okay, consider the content of elements in the body of an average healthy person weighing about 70 kilograms. It has been established that out of 70 kilogram man's weight, oxygen account for 45.5 kilograms, more than half of the total weight. Okay, carbon account for 12.6 kilograms. Hydrogen account for 7 kilograms and nitrogen account for 2.10 kilograms. But phosphorus account for 700 gram. Okay, the total weights of metal in grams. Calcium about 1050 grams, potassium about 140 grams, sodium about 105 grams, magnesium 35 gram, iron 4.2 grams and zinc 2.3 grams. Okay, the content of rest of metal does not exceed 1 gram, particularly copper 0.11 gram and manganese 0.02 grams. Okay, so in human body only 2% metal, but life depends much more upon these elements far more than its figure suggests. Okay, then next we are going to discuss the geochemical effect on the distribution of metals. Okay, all the most essential elements except molybdenum are fairly abundant in the earth crust. Aluminium is about 8.2%, silicon 28.2%, titanium 0.57% and zirconium 0.02% are also abundant, but these are not essential elements because all these forms insoluble oxide at biological pH values and do not form stable complexes with complexing agent of biological significance. That's why aluminium, silicon, titanium and zirconium are not essential elements, because due to the solubility and pH value such as etc. do not form stable complexes with complexing of biological significance, and they do not form stable complexes with biologically significant ligand. Okay, no common element is toxic at level normally encountered, though almost anything can be harmful at too high levels. Okay, all the well-known toxic elements which are currently of much concern in environmental pollution problems are extremely rare in abundance in the earth crust. Okay, for example, arsenic 2 into 10 raised to minus 4%, lead 1.3 into 10 raised to minus 3%, cadmium 2 into 10 raised to minus 5% and mercury 5 into 10 raised to minus 5%. Okay, then classification of elements according to their action in biological system. Okay, next we are going to discuss the classification of elements according to their action in the biological ligands. Okay, elements are classified into 4, that are essential elements, trace elements, non-essential elements and toxic elements. Okay, essential elements are the elements which are essential for the daily nutrient requirement. Okay, which are essential. For example, oxygen, carbon, hydrogen, nitrogen, phosphorus, sodium, potassium, magnesium, chlorine, calcium, sulfur, essential elements. And trace elements are iodine, iron, copper, zinc, manganese, cobalt and molybdenum, etc. Trace elements are also essential but in micro quantity. Okay, that is trace elements. That are iodine, iron, copper, zinc, manganese, cobalt and molybdenum. Then non-essential elements that are not required our body, but that are not toxic. In our body, it's exceeding the requirement level, that may be a chance of toxicity, but the normal level is not toxic our body. That are aluminium, strontium, barium, tin, etc. The next category is toxic elements. In micro quantity, it is very toxic to life or human beings. That are toxic elements: cadmium, lead, mercury, etc. Okay, essential elements are absolutely essential or necessary for life process. Okay, trace elements are also necessary for life process, but non-essential elements are not essential. But if they are absent, or other elements may serve the same functions. Okay, toxic elements disturb the natural function of the biological system. Okay, the toxic elements are disturb the natural functions of the biological system. Okay, next we are going to discuss the metal ions in the biological system, or role of metal in biological system. Okay, metal ions, the excess and deficiency. Okay, concentration of metal ions in human beings system is controlled within very fine limit. Okay, within very fine limit. This control is generally exercised by certain biological complexing agent. The deficiency or excess of metal ion cause disorder, which leads to various disease. Okay, this is the graph showing the deficiency and excess of the metal percentage in our body. Okay, the y-axis is physiological response and the x-axis is the concentration in dose. Okay, then the left side we can say the deficiency of the metal ion. The deficiency cause leading to or deficiency leading to death due to its absence, or deficiency symptom can cause the deficiency. Then the optimal, the center part is the optimum level or the stable state of health. It's the stable or healthy state of health. And the right side or excess concentration or excess dose can cause death or toxic effect. Then first we are going to discuss the calcium. Okay, calcium is a critical element in all human and man, all animals and men. Calcium is a critical element in all animals and men. A healthy human adult has about 1.05 kilogram calcium, of which 99% exists in phosphate, resulting the mineral hydroxyapatite. What is the molecular formula hydroxyapatite? Ca10PO4 6 times OH twice in bone and teeth. Okay, then 99% of calcium exists in phosphate, resembling in the mineral hydroxyapatite in bone and teeth. Okay. The small remainder is in cellular fluids existing in partially ionized or protein bound forms. Okay, the primary dietary source of calcium is milk, 65 to 70%, with smaller amount derived from meat, fish, egg, etc., and still less from non-dietary foods such as nuts, fruits, beans, etc. Okay, the dietary deficiency of calcium is not common problem in nations with high dietary products and protein intake, particularly since the normal individual can regulate intestine absorption and renal conservation mechanism with great precision. Hence, human health problem related to geochemical distribution of calcium, its entry to human food chain and its bioavailability are relatively uncommon. Okay, exemptions include very poor diet such as those low in milk and animal proteins or unusual physiologic other illness such as intestinal metabolism or intestinal malabsorptions. Okay, in case of excess of calcium, it comes into blood as calcium is rejected by cell and its salts are not soluble. Okay, its salts are not soluble. So, excess of calcium leads to the formation of stones, hardening of arteries and cataracts in the eye. Okay, the excess of calcium leads to the formation of stones, hardening of arteries and cataracts in the eye. Okay, then next we are going to discuss the deficiency, the needs and deficiency of magnesium. Magnesium, an abundant element in the earth crust, is a vital to both plant and animals' life. Okay, chlorophyll pigment in plant is a magnesium porphyrin complex. All enzymatic reaction in animals and men that are catalyzed by ATP requires magnesium as a cofactor. Okay, oxidative phosphorylation, DNA transcription, RNA functions, protein synthesis and other critical cell membrane functions are all dependent upon optimal magnesium concentration. Okay, an average man has about 35 gram milligram. Out of it, this 99% is either intracellular or in bone. Of the 60% in bone, 2/3 is slightly incorporated into the mineral lattice, but 1/3 is in a apparently exchangeable bone surface pool. Okay, dietary source high in magnesium includes nuts, seafoods, legumes and vegetables, meat in intermediate in manganese, magnesium content. That's about magnesium. Next, potassium. Okay, potassium or K. An adult human has approximately 140 gram of potassium, of which more than 90% is both intracellular and exchangeable, since muscles contains most of the body intracellular water. It also contains most of the K. Okay, K is the predominant cation in the intracellular water. Okay, since K is found in most animals and vegetable foods, dietary deficiency is exceeding rare except under the unusual condition, such as diet very high in refined sugars, alcoholic individual deriving most of their calorie from low potassium alcoholic beverage in the state starvations, etc. Okay, that's about the sodium. So sodium is essential for the intracellular and extracellular impulse transmission or pH level or the body fluids ion content. Okay, that's about potassium. Then next we are going to discuss the sodium. Sodium is the predominant extracellular cation in animals and man. An adult human has about 105 gram sodium, about 24% located in bone and about 65% in extracellular water. So potassium concentration high in intracellular water and sodium concentration is high in extracellular water. Okay, sodium ion equilibrium is maintained primarily by the kidney, the key organ in water and electrolyte balance. So sodium potassium is essential for electrolyte balance. Okay, sodium chloride, salt is the predominant dietary source. Although the excessive dietary chlorine appear to have no significant ill effect on health. Okay, although the excessive dietary of chlorine appear to have no significant ill effect on health, there is much evidence that excessive sodium intake result in an elevated blood pressure, hypertension, and that reduces sodium intake or increased potassium intake helps to reduce high blood pressure. Okay, that's about sodium. Next we are going to discuss cobalt. Cobalt is an essential element for humans, but its pathway through the food chain to human being remain elusive. Only a little over 1 milligram cobalt is present in an adult human. It is useful to man in so far is unknown, only in the form of vitamin 12 or cobalamin. Okay, vitamin 12 or cobalamin is synthesized only by bacteria. Vitamin enters the human food chain as animal organs or muscles. In man dietary, cobalt deficiency is only likely among strict vegetarian or when the intrinsic factors from the stomach that facilitate B12 absorption is absent or severely decreased, as in pernicious anemia. Okay, that's about cobalt. So cobalt present in vitamin B12, okay, or vitamin B. Okay, next we are going to discuss the zinc. Zinc is the most important element present in metalloenzymes. Okay, zinc. An adult has about 1.5 to 3 grams zinc, with the large amount being in liver and bone. Okay, there is evidence that zinc concentration in blood and several tissue vary considerably in the response to many stimuli. Okay, zinc appears to be critical in many functions, presumably due to the chelation of zinc, as well as during the total parental nutrition, when zinc was not added to the nutritional solution for even a short time as 2 weeks. Okay, a more chronic dietary deficiency zinc combined with other diseases include dwarfism, hypogonism and sexual immaturity. The latter devised with zinc therapy. Okay, dietary source range in zinc concentration from 1400 microgram per gram to 2 microgram per gram or less in fresh fruit and vegetables. Okay, bioavailability of zinc is especially high from animals tissues and low from milk and from grains. Okay, the latter effect apparently due to the binding to phytic acid and fiber. Okay, next we are going to discuss molybdenum. The essentiality of molybdenum in animals and human being assumed to form its presence in the metalloenzyme xanthine oxidase and aldehyde oxidase. Molybdenum is also part of the enzymes sulfate oxidase, inherited deficiency of which cause severe neurological disorder and early death in humans. However, no naturally occurring molybdenum deficiency has been ever documented in animals or man, even though several animal deficiencies have been produced experimentally, particularly by using the molybdenum antagonist. Okay, molybdenum is present in very small quantities. Molybdenum appears to be readily absorbed from gastric intestine tract and excreted primarily through kidneys, though human studies are lacking. Okay, in tissues with higher concentration such as bone, liver and kidney, the molybdenum content can be varied with dietary intake. Okay, there is evidence that dietary molybdenum affect copper metabolism in animals and men. Higher molybdenum intake causing higher intake of molybdenum causing mobilization and excretion of copper. Okay, this effect can be elicited in man even with naturally occurring dietary source of molybdenum. Okay, since molybdenum concentration in grains and vegetables vary enormously and varies with soil content and the possibility of molybdenum induced copper deficiency in man is conceivable though not reported. Okay, then next chromium. The digestion of chromium is as an element essential to animals and man is quite recent. Okay, in so far as is known, the major biological function of chromium is an integral part of organic complex originally isolated from the yeast termed glucose tolerance factor (GTF), glucose tolerance factors. This complex apparently include one chromium 3 ion and two nicotinic acid molecules and may coordinate with 3 amino acid molecules, probably glycine, cysteine, glutamic acid. Okay, experimental data indicate that GTF, glucose tolerance factors, functions in conjunction with insulin and may in fact aid in binding insulin to site of actions. Other activity apparently include a lowering of serum cholesterol and triglycerides. An adult human has about 6 mg chromium. Trivalent chromium is absorbed in the upper gastrointestinal tract, but only in very small amount. Okay, chromium is absorbed in the upper gastrointestinal tract, but only very small amount. Okay, hexavalent chromium is better absorbed, but only trivalent chromium is biologically active as an essential elements. Okay, trivalent chromium as GTF, glucose tolerance factors, is apparently absorbed much better. Thus, conversion to GTF in the gastrointestinal tract may be important and may vary with age of the individual. Okay, chromium in excess amount can be quite toxic depending upon the chemical species of chromium is chromium 3 is much less toxic than the hexavalent forms. Okay, chromium is known a carcinogen and toxic metal present abundantly in tannery effluents in India. As an estimate, about 80 to 90% of the tanneries use chromium as a tanning agent. Of this quantity, the hides take up only 50 to 70%, while the rest discharged as effluents. Okay, that's about chromium. Next, we are going to discuss copper. Then, normal adult human has about 100 to 110 milligram copper. Highest concentration are found liver, kidney, heart and brain. Okay, the prototype functional deficiency of copper in human is an X-linked inherited disorder called Manx syndromes, kinky or steely hair syndromes. Okay, copper insufficiency was first suspected by analogy with abnormal wool in copper deficient sheep. Okay, the defect appeared to be decreased gastrointestinal absorption and or cellular utilization of copper. The essentiality of copper in the consequence of its role metalloenzyme involving several critical biochemical pathways. Several of these enzymes are noted here. Superoxide dismutase, which metabolize the potentially damaging superoxide ions. Okay, lysyl oxide is an monoamine oxidase required for cross-linking collagen and elastin, the structural macromolecules of connective tissue. Okay, dopamine beta hydroxylase, amine oxidase and tyrosinase are all copper containing enzymes that interconvert the major neurotransmitters dopamines. Okay, noradrenalines and adrenaline probable accounting for the high concentration of copper in the brains. The later enzymes tyrosine is also key step in high concentration of copper in the brains. Okay, the later enzyme tyrosinase is also a key step in the pigment production. Okay, cytochrome C oxidase is the key and terminal enzyme of the respiratory chain, accounting for more than 90% of energy muscular contractions. Okay, peroxide, a better known as ceruloplasmin, before its role in mobilization and oxidizing Fe from storage site were recognized. Okay, then ferredoxin is believed to account for 95% serum copper and appears to be a multifunctional protein serving as a major transport system for copper as well. Okay, copper is widely distributed in the food chains, a notable exception being cow milks. Okay, then copper concentration ranges from 20 to 60 microgram per gram in animal tissue to less than 2 microgram per gram in leafy green vegetable and fruits to less than 0.2 microgram per gram in cow milks. Okay, which is much less than in human milks. Okay, in infant especially if premature and not breastfed are therefore most susceptible to dietary deficiency. Okay, excessive loss of copper from gastrointestinal tract due to diarrhea is the most common precipitating factor. Okay, in Wilson disease, copper concentration increase up to 100 times greater than normal. Copper is accumulated in number of tissues, but in particular is found in liver, brain and kidney, which leads to liver and kidney failure and various neurological abnormalities. Death result if condition not recognized and treated. Okay, that's about copper. So, copper is an essential element present in various metalloenzymes too. Okay, then next we are going to discuss iron. Okay, iron also is a trace elements present in hemoglobin, myoglobin, etc. Okay, it's a oxygen transport carriers. Okay, then the average human adult has about 4 to 5 gram Fe. Of this amount, about 60 to 70% present in hemoglobin in the red blood cells. Okay, 3 to 5% in muscle myoglobins. 50% is bound in the Fe storage cellular protein ferritin. 0.2% occur as a component of critical respiratory enzymes, and 0.004% is bound to the serum transport protein transferrin. Okay, so iron present in red blood cells, that is hemoglobins and tissue myoglobins, and ferritin transferrin. Then ferritin is a protein storage code, and transferrin is a iron transfer proteins. Okay, iron deficiency causes anemia because red cells of blood containing less hemoglobin than normal conditions. Okay, acute iron poisoning leads to vomiting, polar shock, circulatory collapse, and coma. Chronic conditions are also known in which iron is deposited in tissue and organs of the body. This condition is known as siderosis. Okay, then next we are going to discuss the toxicity of metals. Okay, the mechanism of the toxicity of metal is very complicated. Generally, toxicity of metals may result from one of the followings. Okay, first one, blocking the essential biological functional groups of biomolecules such as enzymes. Amino acid residue like serine is OH functional groups, cysteine SH group, and histidine N group often constitute the active site of enzymes. Okay, the first factor is blocking of essential biological function group of biomolecules such as OH, SH, N, etc. Okay, a toxic metal ion may bind with these functional groups and block the activity of the enzymes. Okay, then second one, displacing the essential metal ion from biomolecules. A biomolecule with foreign metal ion loses its activity. Okay, due to the presence of these metal ions, the biomolecules lose its activity. Okay, then third factor, modifying the active conformation of biomolecules. Biomolecules are having specific active conformation, and if this active conformation is lost due to the coordination of metal ions, the activity of metal biomolecule is lost. That's are the main toxicity of metals. Then next we are going to discuss the toxicity of metal in the, for example, first one, mercury. Okay, mercury. The source of mercury pollution are industrial waste mining, as mercury is a trace component of many minerals, pesticides, coal, ignite. Okay, containing about 100 ppb of parts per billion of Hg. It is well known toxic metal came to limelight after the incident of Minamata disease in 1953 to 60 in Japan. Okay, Minamata Bay is a disease caused due to the excess mercury. Okay, 111 cases of mercury poisoning were reported who had eaten mercury containing fish from Minamata Bay. Among them, 45 people dead. The sea fish were found to be containing 27 to 102 ppm parts per million of Hg in the form of methyl mercury. The mercury source was effluent Hg containing catalyst waste from vinyl chloride plant. The mercury source was the effluent from vinyl chloride plant, Minamata Chemical Company, M.Sc, releasing into the bay. This was followed by more tragic report of mercury poisoning from Iraq in 1972, where 450 villagers died after eating wheat which had been dusted with mercury containing pesticides. These two tragic events boosted the awareness of mercury as pollutant, and ultimately resulting in its being studied more extensively than any other trace elements. Mercury can be toxic by ingestion or inhalation, but the toxicity depends upon the chemical forms. Soluble inorganic mercury salt are highly toxic. In excess, HgCl2 causes corrosion of the intestinal tract, kidney failure and ultimate death. Okay, then reason for toxicity. Toxicity mercury is based on the strong affinity, very high formation constant between 10 raised to 6 to and 10 raised to 22. Okay, the toxicity of mercury is based on the strong affinity for deprotonated form of thiol ligand such as cysteines. Therefore, thiol RSH with sulfur hydryl group SH are also called mercaptanes, mercurium captains. So mercuric bind strongly with thiol group of protein and enzymes, and this binding changes the conformation of protein about active sites. Mercury is a soft acid. S of SH is a soft base, so strong interaction between mercury and SH group can be explained on the basis of stronger soft binding or soft interactions. Okay, that's about mercury. Next, we are going to discuss the toxicity of cadmium. The source of cadmium pollution in urban area are metallurgical plant, cadmium plating and battery fabricators. Acute cadmium poisoning leads to nausea, salivation, vomiting, diarrhea and abdominal pain. Cadmium deposition tends to cumulative in the kidney with lower concentration in the liver. Okay, another characteristic of cadmium poisoning is brittleness of bones. Okay, cadmium occurs in natural association with zinc minerals. A severe outbreak of chronic cadmium poisoning occurred along the Jinsu river of northwest Japan and was known as Itai or Ochu disease. Over 20 years period, around 100 people died. In 1961, cadmium was found to be the cause. Okay, the region was due to old disused zinc mine and river water which containing cadmium used for irrigation of rice, and people died after eating the rice. Okay, the reason for the toxicity of cadmium. Next we are going to discuss the reason for the toxicity of cadmium. Cadmium similar to zinc, therefore cadmium too can displace zinc in many zinc enzymes. Like mercury, cadmium too also bind strongly with the SH group of cysteine residue of enzymes. Example, carbonic anhydrase, dipeptidase, carboxypeptidase, etc. Cadmium too, like other toxic metal ions, affect the active conformation of biomolecules due to the strong binding. Okay, cadmium also change the active conformation of biomolecules. Okay, that's about cadmium. Next we are going to discuss the lead. The source of lead pollution. Battery industry is the largest single user of lead. But the leaded petrol account for more than 20% of the total lead consumed per year, and 90% of the lead released to atmosphere is from gasoline exhaust. Okay. Triethyl lead cations C2H5 thrice Pb plus is formed from the tetraethyl lead by the dissociation of carbanions. The toxicity of this organometallic cation result from the permeability of membrane, including the very discrimination blood-brain barrier, causing several disorder of central and peripheral nervous system. For example, cramps, paralysis or loss of coordination, etc. One of the characteristic symptoms of lead poisoning is anemia. Okay, then next, reason for toxicity. Like mercury and cadmium, lead inhibit the SH enzyme but less strongly. Major biochemical effect of lead is its interfere with heme synthesis by inhibiting several of key enzymes involved in the overall process of heme synthesis. Okay, then next we are going to discuss the use of chelating agent medicine or chelation therapy. treatment of metal poisoning or detoxification by chelating agent. So many chelating agent which is used to treat metal poisoning. The mechanism behind this treating is these chelating agent which are non-toxic, which chelate the excess metal present in our body. Okay, the treatment of metal poisoning is done by the use of chelating agent. Then mostly the treatment of metal poisoning is done by the use of chelating agent. It is hoped that chelating agent will form soluble, stable and non-toxic complex which are readily excreted. Okay, then the criteria for potential chelating drug. The criteria for a potential chelating drugs are: first one, it must bind the metal strongly to complete for it with biological ligand and excreting as a soluble chelate. Okay, then it must bind the metal strongly to complete for it with biological ligand and excreting as a soluble ligand. Okay, that's the first point. Then second one, it should be selective for the metal ions, or it should be selective for the excess ions. If it is non-selective, then there will be a harmful side effect from the removal of other metals, particularly calcium and zinc from the body. Okay, that means the ligand should be selective. Okay, then the chelate must be low toxicity and not metabolized. Okay, it should remain unchanged in biological system. Okay, the chelate itself as non-toxic. It should be non-toxic. Okay, then the fourth point, it should be capable of penetrating into the metal storage sites. Okay, fourth point is it should be capable of penetrating into the metal storage sites. Then the fifth one, chelate should be less toxic than the free metal ions. Okay, chelate should be less toxic than the free metal ions. Okay, a suitable multi-ligand that can satisfy all the coordination position of the metal ion would be ideally suited in the elimination of the metal ions. So the chelated metal ion cannot bind any binding site of enzymes and proteins. Example is EDTA. EDTA is the most familiar example of chelating agent used in the chelation therapy. So the first chelating agent used in the chelation therapy are ADTA, ethylene diamine tetraacetic acid. Okay, next one is British anti-Lewisite or BAL. British anti-Lewisite. The short form of British anti-Lewisite is BAL, or 2,3-dimercaptol 1-propanol. 2,3-dimercaptol 1-propanol. Okay, then next one, D-penicillamine. D-penicillamine, it is used for urinary excretion of copper in Wilson disease. Okay, it is the D-penicillamine, it is used for urinary excretion of copper in Wilson disease. D-penicillamine, which contain a OH and SH-NH2 functional groups, but BAL contain a SH and 2 SH and 1 OH groups. Okay, then next we are going to discuss drug appear into involving interaction with metal ions. Drugs appear to involve interaction with metal ions. Many drugs are suspected of acting by chelations. Okay, some examples are diuretics. Acetazolamide these are the drugs that promote the formation of urine. For example, dicarb or acetazolamide. Acetazolamide.